Hi, I'm Akshita.
Malia?
I'm Anirudh.
I'm Arushi.
I'm Aryan.
I'm Catherine.
And I'm Sunandita.
And this is our project investigating the effects of curcumin and its analogs on alpha-synuclein aggregation in Parkinson's disease. And our advisor is Ms. Carly Truong. So Parkinson's disease is one of the most common neurodegenerative movement disorders. And Parkinson's disease has affected over 10 million people worldwide. The causes include both genetic and environmental factors.
So the scientific reason is with our brain, and we have dopaminergic neurons which produce dopamine. However, the area of the brain, the substantia nigra, which produces this dopamine, stops producing it. Without this dopamine, it will lead to many symptoms such as problems with moving muscles and regulation in emotions.
Alpha-synuclein is a natively unfolded protein encoded by the SNCA gene that contains 140 amino acids. The normal function of alpha-synuclein is not well understood in the healthy brain, but research has shown that the misfolding of this protein is a significant factor in Parkinson's disease. More specifically, the accumulation of these misfolded alpha-synuclein proteins forms Lewy bodies, which interfere with the neural function and contribute to the degeneration of dopamine-producing neurons.
Next, I will be talking about this structure. The structure of alpha-synuclein is composed of three regions: the N-terminal, the C-terminal, and a central region, also known as the NAC domain, which stands for non-amyloid beta component. The NAC domain is particularly important because it is highly hydrophobic. This makes it responsible for the aggregation and misfolding of alpha-synuclein.
In terms of alpha-synuclein aggregation, the SNCA gene codes for alpha-synuclein. Essentially, an unfolded monomer will become a misfolded protein due to a mutation in the gene. The monomer is unfolded and soluble, while the misfolded proteins form from beta sheets, which are folded randomly and are insoluble.
What is curcumin? Curcumin is a polyphenol group compound common in South and East Asian cooking, which is derived from turmeric. Recent research studies have shown that natural polyphenols like curcumin have potential in treating neurodegenerative diseases like Parkinson's disease due to their therapeutic properties. Curcumin is a plant-based molecule with antioxidant properties and anti-inflammatory properties, which can inhibit oxidation, which contributes to the dopamine cell degeneration. Additionally, curcumin is a small molecule which allows it to cross the blood-brain barrier.
Currently, we are working with alpha-synuclein monomers and are beginning to induce them into their fibril form so we can assess its aggregation. This is a 7-day process where the monomers are essentially agitated in a shaking incubator for 7 days. Once the fibrils form, we will determine the fibril formation via a fluorescence assay. We are currently discussing ANS fluorescence, THT, or THS fluorescence. In the future, once we apply curcumin as a treatment from its analogs, we will check its binding affinity to alpha-synuclein via a Nile red staining, which highlights the areas in which curcumin did not bind. These are the current and future procedures we are and will be carrying out for our project. Here we are monitoring our fibrils. We've been doing this for the past week to ensure quality. We want to thank our advisor, Ms. Carly Truong, and the ASDRP Biological and Life Science Department for making our research possible. Thank you.